Traveling through the years of the coronavirus pandemic, we found that there have constantly been new novel SARS-CoV-2 variants that have emerged. There are several hypotheses out there that explain how these variants can actually emerge. And one of these hypotheses is chronic infection in individuals that are unable to clear viral infections. And to this group, immunosuppressed individuals belong. And immunosuppression, for example, can appear after solid organ transplantation when these people receive immunosuppressive medication. And one of these medications is mycophenolic acid, MPA. And surprisingly, we found that MPA can actually, in a dose-dependent manner, inhibit a variety of different viruses, including 229E and endemic coronavirus, but also other respiratory viruses like influenza A virus and the respiratory syncytial virus. And we also found that hepatitis E, but also Mpox virus can be inhibited by MPA. And when doing mechanistic studies, we found that MPA actually inhibits virus replication. And as we realized that these viruses, all these different viruses, have very different replication cycles, we also thought that the mode of action might rather be a generic process than a specific interaction pattern or protein. And in the literature, we found that MPA is a non-competitive inhibitor of the inosine monophosphate dehydrogenase that converts IMP to XMP, and then further down the line, GTP is produced that is necessary for replication of RNA and DNA. And by this inhibition, we can actually deplete GTP from the cellular environment and thereby impair virus replication. And to test for this hypothesis, we can, while treating with MPA, ectopically substitute the GMP, which is further downstream of the enzyme, to restore GTP pools and then restore also virus replication. And this is exactly what we tested for. While treating the cells with MPA with a suboptimal concentration of MPA and substituting different concentrations of GMP, we found that viral titers can actually be restored, whereas when we tested for the different other monophosphates, this effect was not observed. And because we also thought that this mode of action might give rise to novel SARS-CoV-2 variants, we also passaged the virus in the presence of MPA, tacrolimus, and MPA, tacrolimus, and prednisolone, because these drug combinations are most predominantly used in the clinics after solid organ transplantation, and we found that the virus can actually adapt to this treatment. And we also found that the phenotype of the plaque sizes changed. And when doing RNA sequencing, we found that actually by the time the virus adapts to the MPA treatment, 3 different mutations have arisen that when generated by reverse genetics, we found that the triple variants or the combination of these 3 variants can actually yield to higher viral titers in the same time frame compared to the wild type and the single variants. What is important to say is that these variants remain susceptible to clinically relevant drugs such as molnupiravir, Paxlovid, and also remdesivir, and they are also susceptible to neutralization from vaccine-induced and also infection-induced antibodies. And to sum this up, MPA, although it has an initial antiviral effect, may lead to the emergence of novel SARS-CoV-2 variants, which eventually have an altered viral fitness. And what I didn't tell you before is that these mutations can also be found in vivo, and this means that these in vitro adaptation studies are a powerful tool to actually predict the emergence of virus variants and also evaluate the pandemic potential of these variants. And this also means that monitoring chronically infected patients does make sense to monitor also the emergence of novel SARS-CoV-2 variants and other viruses.